Hello. In this video, we're going to talk about the details of protein synthesis. So, that is how a protein actually gets made from DNA. So, if you recall, proteins are super important for different cells to do their jobs. So, depending on the type of cell and the type of living thing, that cell is going to have a different set of proteins. And if you recall way back, proteins are also known as polypeptides, and they are made up of amino acids. So, amino acids are these little pieces that get linked together to form polypeptides. And polypeptides get folded up into unique three-dimensional shapes. And what we call essentially a protein. It is the order or the sequence of those amino acids in a polypeptide that gives that polypeptide or that protein its three-dimensional shape and its really important properties. So in this little picture example down here, say there's this is a chromosome and then a little piece of DNA within that chromosome right here. This DNA sequence contains the gene for a specific flower color. That DNA sequence gets kind of like highlighted, if you will, because it's important. And it gets highlighted, and then what we call that gene is expressed, which means through the process of protein synthesis, the expression of that gene is purple flowers. Okay, so keeping all that in mind, how the heck does all this happen? Well, there's this idea in biology that we call the central dogma. And the central dogma is DNA contains information, really important information about genes. These little specific genes contain that important information, and these genes need to get expressed. So how do they get expressed? By taking that information from the DNA, storing that information, that message, in what is called messenger RNA, and then taking that message and translating it into a protein or a polypeptide. So DNA to RNA to protein or polypeptide. That is what we call the central dogma. And there are two specific processes that allow this to happen. So again, here's our DNA. DNA goes through a process of transcription, and it is transcribed, like kind of written down in RNA, messenger RNA specifically. And then it is translated, just like one language to another language, it is translated into a protein. So the whole time, DNA has the message in it, but the message can't get out without RNA. And again, these two processes are called transcription and then translation. So we're going to start by talking about transcription. So transcription happens in the nucleus. Everybody see that? The nucleus. In the nucleus, we have our DNA. DNA is extremely important. We have a molecule called RNA polymerase, and RNA polymerase binds to DNA. Again, that DNA is in the nucleus, and it separates the DNA. It untwists it, and that RNA polymerase knows where to bind. It finds these special regions that contain genes. So no, the RNA polymerase doesn't bind just anywhere on the DNA. RNA polymerase knows where to bind, and then it finds those spots because it knows where the genes are located. So here's our DNA. So it's a twisted ladder, and in the section of the DNA where the gene is located, that little section untwists. and separates. So here we're focusing on this piece of DNA here. And we know this is DNA because it has T in it. A, T, G, C, T, A, G, G, C. Remember, DNA has the bases A, T, C, and G. However, RNA doesn't have T. So we know that this is RNA here coming in and now binding to this section of DNA that contains the gene. So this section of DNA has the gene, and we need mRNA to get that message out. So the DNA A binds with U. Again, there's no T. T binds with A, G binds with C, C binds with G, T binds with A, so on and so forth, to create our final mRNA strand. Again, it's mRNA, so it's a single strand. And again, this is all happening in the nucleus. Here's just another picture of this process. It's a different cartoon version. We have our DNA double helix here. The DNA is opening up, unwinding and opening up here. Here is our DNA, G, A, T, C, A, T, and then here's our mRNA complementary binding with our DNA, C, U, A, G, U. And that mRNA, once it has been created, there might need to be little modifications. We're not really concerned with that at this point, but once it's all done and the mRNA is processed, that mRNA floats out of the nucleus and it finds a ribosome. Remember, ribosomes are protein-making little factories. They're typically represented as little circles. And this is where translation happens. So again, we've written down the message in transcription. That messenger RNA has now left the nucleus, and it is time for translation to happen, just like translating from one language to another. So during translation, again, this happens on a ribosome. Amino acids are brought to the ribosome by tRNA. Okay, so tRNA, little person, has carries with it an amino acid. And the amino acid order or sequence is very important. And that sequence or order is determined by the order of the mRNA. Okay, that determines how the tRNA is going to bind and what amino acid gets carried along. So I'm skipping along to this picture because it's fine in words, but it's really nice to show a picture. So during translation, here we have our ribosome. This is mRNA. And then this thing here is our tRNA. And again, I call it a tRNA little person because this is like the head of the person, the amino acid, and then these are like little arms, and then it's like 3 little feet. There are some specific names here that I just want to point out to you. So again, this is the tRNA. The little head of the tRNA person is the amino acid. And that tRNA brings the amino acid over to the ribosome, and it binds to the mRNA right here. So let me just highlight and show you right here. This is very important. So this little group of 3, 1, 2, 3 on the mRNA, that is called a codon. And that codon indicates the amino acid that the tRNA brings. It's pretty cool, but it's extremely important for making sure that the protein that's created is the correct protein. Here is just in this next slide here, I'm showing you a little bit more detail of the tRNA. You don't have to memorize this or anything, but I just think this is kind of interesting. So yes, it's shaped like we represent it in cartoons as this like little person with the amino acid attached at the top here, and then the arms, and then along the bottom, there's this little group of 3 that binds to the mRNA codon. We call that the anticodon. And then in this picture, we see that anticodon here. And then this is really just your RNA folded up, your single strand of RNA folded up into that shape. Okay. So again, that is transcription and translation. And these tRNAs just keep going until they are told to stop. Okay. If you have any questions, feel free to reach out. I highly encourage you to write this down, watch all the videos in order, and I think that's going to really, really help you with this process.